Good morning. Today's topic is membrane transport. The learning objectives of today's topic are: describe transport mechanisms across the cell membrane, describe transport of ions and small molecules from one cell to another cell. So these are the learning objectives of today's topic. The transport mechanism across the cell membrane are broadly divided into three different types. They are passive transport, active transport and vesicular transport. The flow chart of membrane transport is in this way, that is, it is divided into passive transport, active transport and vesicular transport. Passive transport is a type of transport which occurs without involvement of the energy that is produced by the hydrolysis of ATP. Active transport is the type of transport which occurs with the involvement of energy that is produced by the hydrolysis of ATP. Vesicular transport occurs by endocytosis and exocytosis. So let us see one by one. The passive transport, the examples are simple diffusion, facilitated diffusion and osmosis. Active transport, which occurs by the hydrolysis of ATP, which requires energy by the hydrolysis of ATP, is of two types, that is primary active transport and secondary active transport. And the vesicular transport is endocytosis and exocytosis. Let us see them in detail one by one. The first one is passive transport. Passive transport is a type of transport that is downhill. So passive transport occurs from high concentration to low concentration. The substances passes through the membrane from both sides. However, net movement occurs down the electrochemical gradient. Transport does not require energy in the form of ATP. So the passive transport doesn't require energy in the form of ATP. Important point to remember. Then the examples of passive transport are simple diffusion, facilitated diffusion and osmosis. So simple diffusion. Simple diffusion is the process of passive transport in which the molecules move from an area of high concentration to an area of low concentration. The energy that causes diffusion is derived by the kinetic energy generated due to the random motion of the molecules. The examples of substances that pass through the cell membrane by simple diffusion are the transport of gases like oxygen, carbon dioxide and urea, ammonia and other ions by the process of simple diffusion. Then simple diffusion can occur through the cell membranes by two different pathways. In one pathway, the foreign particle itself or the foreign diffusing substance itself is lipid soluble and it enters through the interstices of the lipid bilayer by diffusing directly, as it is lipid soluble foreign particle. And the second pathway is through the watery aqueous channels which are formed by the transmembrane proteins. The next one is facilitated diffusion. This is also called the carrier-mediated diffusion, as the process of diffusion is facilitated by the carrier protein in the membrane. There are many types of carrier proteins in the membranes, each type having binding sites that are specific for a particular substance. So each carrier protein has a specific binding site for a particular substance only. That substance only can bind at a particular binding site of a carrier protein. No other substance can bind at that particular site of the carrier protein. So then the glucose and the most of the amino acid cross the cell membrane by the process facilitated diffusion. Like simple diffusion, facilitated diffusion is also a downhill transport that requires energy. Facilitated diffusion is more rapid than simple diffusion. These diffusion processes are not coupled to the movement of other ions. They are known as uniport transport process. So facilitated diffusion is also known as uniport transport process. In osmosis, osmosis is the process of movement. It can be defined as a process of movement of water from solution with the lower concentration of solutes to the solution with the higher concentration of solute when both the solutions are separated by a semi-permeable membrane. Then the second one is active transport. Active transport differs from the passive transport in utilization of energy. So the active transport is also called as uphill transport because the transport occurs from lower concentration to higher concentration. Transport occurs against the electrochemical gradient of the substances. So that is called as uphill transport. So active transport requires energy, and the energy utilized for the transport is derived by the breakdown of ATP. Active transport exhibit saturation kinetics. So this is because of limitation in the rate of availability of carriers or supply of energy. So active transport is an uphill transport which occurs against the electrochemical gradient, and it requires energy in the form of ATP, and it exhibits saturation kinetics in the rate of availability of carriers or supply of energy. The active transport mechanisms are of two types, that is primary active transport mechanism and secondary active transport mechanism, which we will learn in detail. So primary active transport mechanism. So primary active transport is a transport mechanism that directly utilizes energy derived from the hydrolysis of ATP to ADP. The mechanism is operated by ion pump. The diagram here presented to you shows that the sodium, which is present intracellularly, is transported to the extracellular out of the cell by utilizing ATP and a carrier protein. So this type of transport where the sodium is pumped out of the cell by utilizing ATP is called as active transport, and more accurately it can be called as a primary active transport, as the sodium is pumped from low concentration to the high concentration by utilizing energy. So these are the examples of ATP-dependent active transporters present at different locations. The examples may be the sodium potassium ATPase, calcium ATPase, etc. etc. Then classes of transport systems involved in facilitated diffusion and also active transport. The carrier proteins of facilitated diffusion and active transport can be classified according to the number of molecules transported and the direction of movement. A uniport system transports only one type of molecule bidirectionally. For example, sodium channels transport sodium, potassium channels transport potassium. Transport of glucose by facilitated diffusion are the examples for uniport transport systems. The second one is the Symport system. The two substances are moved simultaneously in the same direction. For example, transport of sodium and glucose from the lumen of the intestine into the epithelial cell. In the same way, the transport of sodium and amino acid from the lumen of the intestine into the epithelial cell. So this is a good example or clear-cut example for secondary active transport, where the sodium which has come to the extracellular fluid by active transport is being utilized by the glucose to get into the cell utilizing a carrier protein. So this Symport system is a clear-cut example of secondary active transport. The third one is an Antiport system. The two substances are moved simultaneously in the opposite direction. A typical Antiport carrier is the sodium-potassium pump and sodium-hydrogen exchange found in the proximal tubular cells of the nephron, and sodium-calcium transporter is also an example of Antiport carriers. Here one ion enters into the cell with exchange of another ion that is exported out of the cell. So this type of transport system is called as Antiport system. That is, one ion is entering into the cell, and for replacement of this, one ion is exported out of the cell. So an Antiport system. So here the diagrammatic representation of sodium potassium pump, where you can see the sodium which is present inside the cell is pumped out of the cell, and potassium which is outside the cell is pumped into the cell. In this process, ATP is hydrolyzed and energy is gained, or energy is required or energy is utilized by hydrolyzing ATP in presence of enzyme sodium potassium ATPase. So it is a good example of antiport system. The secondary active transport. The transport depends on primary active transport of sodium by sodium potassium pump. It is known as secondary active transport. Examples of secondary active transport is reabsorption of glucose from the intestine and kidney tubules across intestinal and renal epithelial cells. So the secondary active transport is not an independent one. So it is actually dependent on the primary active transport system. So how it is dependent? It is dependent where the primary active transport, where the sodium is pumped out by the primary active transport, is being utilized in the secondary active transport system for pumping of glucose into the cell by utilizing sodium and a carrier. So secondary active transport. So this is how the diagrammatic representation of secondary active transport looks like. So here S1 is primary active transport and S2 is the secondary active transport. So S1, by utilizing the energy produced by hydrolyzing of ATP and a carrier protein, it is pumped out of the cell. So it is a primary active transport mechanism. So once the sodium S1 may be considered equally with sodium. So S1 or sodium, which is present extracellularly and which is the result of the primary active transport, is being utilized by the secondary active transport system. So S2 may be considered equal with glucose or amino acid, where the S2 along with S1. So S1 is sodium, S2 is glucose or amino acid. Yes, sodium along with the glucose enters into the cell, that is absorption without utilizing any energy. So secondary active transport occurs without utilization of any energy, but it is dependent on the primary active transport, where the sodium is pumped out of the cell, where S1 is pumped out of the cell by utilizing hydrolyzing the ATP. So secondary active transport system. Then the differences between passive and active transport. The passive transport occurs along with the electrochemical gradient. Active transport occurs against the electrochemical gradient. And passive transport, there is no need of utilization of energy. Active transport, the utilization of energy occurs by hydrolysis of ATP. The passive transport, the saturation kinetics is not exhibited, and the active transport, the saturation kinetics is exhibited. So this is the major differences between the passive and active transport systems. Then the last and final one, that is vesicular transport. The vesicular transport is special for macromolecules. This transport has no relation with the concentration gradient. However, it may utilize the metabolic energy. Vesicular transport mechanism are of two types, that is endocytosis and exocytosis. Let us see one by one in detail. So endocytosis. Endocytosis is the process of transport in which a substance is taken into the cell by means of vesicle formation. Endocytosis occurs by two mechanisms, that is constitutive endocytosis and clathrin mediated endocytosis. So what are these two? Let us see in detail. So this is the diagram representing constitutive endocytosis, where the macromolecules which comes near the cell membrane, there is the formation of endocytic vesicle in the cell membrane, and the macromolecules are dragged inside the vesicle, and there is separation of vesicle from the cellular membrane, and this is how the macromolecules are absorbed into the cell by forming endocytosis. And this is the example for clathrin mediated endocytosis. The clathrin have got receptors which are specific for some specific macromolecules. So the specific macromolecules, which when it comes in contact with these receptors, they get absorbed to these receptors, and the attachment, they get attached to these receptors. The attachment of macromolecules to the receptors that is to be absorbed, Then these receptors along these macromolecules along with the receptors undergoes invagination to form an entire pit. Then breaking of invaginated portion of the membrane and formation of endocytic vesicle occurs. So this is how the clathrin mediated endocytosis occur. Then exocytosis. Exocytosis is the release of macromolecules from the cell to the exterior, which is reverse of endocytosis. By exocytosis, the hormones, neurotransmitters, digestive enzymes and undigested foreign particles are released from the cell to the exterior. Amniocytosis is the term refers to exocytosis of specific hormones and granules. So this is the diagram which represents exocytosis. The cell membrane, so which the macromolecules which are not required by the cell, they form exocytic vesicle, and this exocytic vesicle comes near to the cell membrane and get fuses with the cell membrane and then discharge the contents of vesicles into the extracellular space. This is how the exocytosis occur. So this ends your topic, and after this you will be given assignment. So please write the assignment, complete the assignment and send to the email address given in this video. Thank you.